Hi everyone. Grüezi miteinander und Sali zusammen. My pleasure to welcome you to Basel, also in a sunny evening. It's a terrible conference, isn't it? We have too much to do, too much excitement. So we'll have hopefully a relaxing and interesting session. My name is Pete Seiler. I'm from INCATE, from the Incubator of Antibacterial Therapies in Europe. We're very happy to foster that session. We are supporting young stage innovators on their way to become investable ventures. And you're invited to see more of how we do that at booth number 9. I'm sure you have seen that. It's a very prominent booth. We also have a pitch competition today at 6 p.m. where you can see a bit what we do. And of course, please also come to the poster award. So we're also responsible for organizing that. We have a genuine interest in non-traditional assets. You will see that in a minute. And we want to learn a bit about your interest, about the risk we go with that, or the opportunity we do. So it's also a bit an exam to all the audience here. We want to get a good journey through all what we can learn, to offer some information to our ventures and to you, the audience, but also to learn what is the right way to go. We have an excellent opportunity to foster that discussions with a journey from getting an overview on the field, seeing one potential value proposition, and then go a bit into regulatory, and then end up with one brave example who went through all that with a non-traditional asset or non-traditional antimicrobial, or in short, NTA. I will start and keep it short here. We'll have four presentations with short questions in between. However, if they're too burning, we go with the questions after each session and go on like that. I'd be happy to start with Valeria, and all the bios are so impressive that I need help here to read. Thanks, Valeria, for coming. Valeria is a clinical pharmacologist with 18 years of experience in research and development, but also in medicines, regulations, and public health policy. She joined WHO in 2017, where she's leading the global AMR R&D and innovation portfolio. So it's a perfect person to give us an overview of what is going on. In her role, she coordinates priority setting and partnerships to advance novel antimicrobials. And we are very grateful to have you here, Valeria, to give us the overview. Thank you very much for coming.
Very kind introduction, and thank you for organizing actually this panel that gives us the opportunity to see a little bit more about NTAs. I will be talking about NTAs in clinical development today, intended for commercial use, of course, that are addressing bacterial priority pathogens. And together we will see some trends and opportunity. This is my disclaimer slide. I will start presenting the definition of NTAs, how they propose them with a strategic differentiation, how they align with global funding priorities, and we will conclude seeing with some pipeline dynamics and the path moving forward to shift from challenges to opportunity for collaboration. So NTAs are really an heterogeneous group of strategies and therapy that are intended to improve treatment outcome in drug-resistant infection, being used as add-on or standalone therapies. Today, we don't have a universally accepted definition of NTA yet. We define them usually as everything else that is different from traditional small molecules, and that they imply that use an indirect mechanism of action, such as biofilm disruption of antivirulence factors. NTA are a core part of the AMR toolkit, with antibiotics, with vaccine, with diagnostics. WHO classified them in 6 categories. So NTAs that recently entered the clinical and preclinical development are classified as monoclonal antibodies, antivirulence factor, phages, immune and microbiome modulating agents, and then we have a basket with many different modalities. So what we can see here very clearly is that there is really a substantial diversity in the approach that are being explored. This is why for NTA we cannot adopt a single translational pathway. NTA are also central to the AMR innovation agenda because they contribute to member states' strategic armamentarium of medical countermeasures. In 2024, global leaders signed the political declaration of the UN high-level meeting on AMR, directly calling for alternatives to antibiotics. And this declaration was signed by 193 member states. NTAs are also part of the WHO strategic R&D agenda. WHO revised NTA in clinical and preclinical development since 2019. And the WHO Advisory Group on R&D also provided some recommendations to strengthen our so fragile and thin pipeline. And one of these recommendations was to expand R&D towards non-traditional approach, recognizing also that the development of NTA is a relatively new endeavor for everyone. So we're really learning from each other. NTA also align with the global funders' priorities, because they address some scientific gaps that small molecules cannot address. And they respond also to the global health needs that global funders increasingly recognize. NTA provide a strategic differentiation beyond bacterial killing and growth inhibition. For instance, they target biofilm, they target intracellular persistence, virulence. They provide also a precision approach, slowing the emergence of resistance or minimize collateral damage. And some of them have also the potential to restore antibiotic efficacy. Global funders also recognize the critical importance of adopting an holistic approach to their health security portfolio of products. Small molecules alone cannot address exhaustively all the bacterial priority pathogens. Even though funding calls strategically shift according to gaps and needs, the overall funders' missions include vaccine, diagnostics, antibiotics, and NTA. And also prioritizing equity and access, NTAs could represent a sustainable solution for LMIC healthcare system. So the bottom line here is that NTAs align with funders' priorities, not by the modality that they represent, but by the problem that they contribute to solve. And we have also a proven investment shift. And here, thanks to our partners in Kate, Carbex, AMR Action Fund, I have some example on how their portfolio is composed today. For instance, more than 50% in Kate portfolio is represented by NTA, and there is a focus on antivirulence and phages. For Carbex, more than 33% of the portfolio is represented by NTA, with a focus on CRISPR, immunotherapy, phages. And for the AMR Action Fund, we also have a similar percentage, more than 30%, with a focus on phages, peptides, and microbiome. And here I want to present you some success story. So in 2022 and in 2023, we had the first approvals of NTA. They are all live biotherapeutic products intended for a recurrent C. diff infection, and they were the first agents the first ever NTA authorized. C. diff is not any longer on the bacterial priority pathogens, but is reviewed by products against C. diff are reviewed within our pipeline analysis because it's an important pathogen linked to, for instance, colorectal cancer. We also have another product, an antivirulence factor approved in the Russian Federation in 2024 for critical and high priority pathogens. Let's see now some pipeline trends. So the number of antibiotics declined of 10% since 2023, while the number of NTA increased of 5-fold from 2019 to 2025. And today we have 40 non-traditional agents in clinical development. As for antibiotics, most of NTAs today are in phase 1, phase 2 of clinical development. We have few agents in phase 3, and we don't have any agent in under regulatory review today. Traditional antibiotics target approximately for half of them target critical priorities, while NTA focus mostly on high and medium priorities. We have 27 agents that target bacterial priority pathogens, 11 agents that target C. diff, and we have also 2 agents intended for TB. But let's see a little bit more about the activities of these 27 compounds against the bacterial priority pathogens. We have 4 agents targeting E. coli, 5 agents that are broad spectrum, gram-positive, gram-negative, and we have 19 agents that target the critical and high priority, with a focus especially on Pseudomonas and MRSA. And it's really important to know that while we have these 5 broad spectrum agents, so potentially we have some candidates targeting critical priorities, we don't have any single agent in clinical development intended for Acinetobacter, that is a critical pathogen responsible for serious infections, outbreaks, especially in ICU. If we look now at the categories, phages are dominating the space with more than 50%. We have 14 compounds in clinical development. We have generally quite a great safety confidence. However, we see some challenges to overcome phase 2-3 studies. The second most represented categories is antibodies. Most of them target MRSA, and we see some challenges in development related to immunogenicity. And if we look at the approaches, most of the NTAs in clinical development are intended in combination with traditional antibiotics, while 10 of them are intended as standalone agents. However, all these 10 agents are intended as preventative medicine. This complicates a little bit clinical trial design, and also there are challenges in identifying the target population. And let's conclude looking at what is really needed to expand the R&D for non-traditional agents, as our advisory group recommended. And I wish to talk a little bit of the financial landscape that is underpinning our scientific efforts. And these data were produced by the Global R&D Hub and in collaboration with Lloyd Capesti. So the funding for antibiotics have been fluctuating over the years, reaching a peak in 2020 and declining since then. While we see that for non-traditional agents, they maintain a more steadier and resilient baseline. And this probably shows the interest in the space from global funders. And let's see now what is taking to enable this shift from challenges to opportunity. First is about strategic positioning. Probably we should stop calling NTA as alternatives to antibiotics. They represent today a critical evolution of the AMR toolkit. Second, we need a robust characterization early on. Academia, micro-size companies are driving our pipeline today. However, we need an alignment. We need to bridge expertise with the preclinical and clinical experience. Third, we need a scientific precision. Often we see too promising in vitro data that fail to demonstrate the clinical benefit. So what is needed is to move towards validated physiological models and to try to define convincing, clear clinical benefit, and we will hear from Nia much more about it. Only in this way we can flip the complexity of NTA into really a tool of precision medicine. And the last success is measured by access. Standardizing manufacturing, leveraging on global interest, we can ensure that this innovation reach every patient everywhere. So only turning challenges into potential opportunity for collaboration, we can really shape a roadmap for success, for better science. We can reduce failure rates, and we can make sure that these fields becomes more attractive for long-term investors. This is the very end of my presentation, but before concluding, I would really acknowledge all the expert organization that supported the WHO work, and I would really also acknowledge the generous contribution of the European Commission, ERA, for enabling this and also the broader R&D portfolio. So thank you all for listening.
Terrific. Thank you very much, Valeria. It is open for questions now. Yes, we get. Valeria, I said I allow you to choose, but let's go over here. Yes. Oh, I thought it's you who. You were just moving the mic. Okay, good. It's also fine. Who would like to get the mic? While you are searching for interested parties, Valeria, do you think we have to, you started with subcategorizing the NTAs. Is that key that we do that we shouldn't call them just NTA, so that we can unlock maybe at least one more potential?
Thank you, Peter. This is really a good point. It's really just a subcategorization that we adopt to try to see what were like the most represented categories, for instance, for phages, but it was a categorization that was done for like internal purpose to see how the space is moving. I think it's also important to know to see what kind of products are like receiving most like interest from the scientific community.
Thanks.
Thank you.
Yes, we have a question over here. Too early.
Hi, really, thank you so much for the presentation. My question is more towards how do you align with the diagnostics for NTA, because all of the ones that you have mentioned, for example, if it is antivirulence strategy, to effectively prove that in a clinical setting, it needs to be having an adjacent, diagnostics. How do you look at that?
So thank you for this important point. At the beginning of my presentation, I really tried to make sure that the message was clear. For the AMR response, it's really important that we look at the whole aspect, from the R&D side at least, that we look at vaccine and diagnostics, therapeutic solutions that are antibiotics, that are non-NTA. Within WHO, we are leading several initiatives around diagnostics. Personally, and with many of the experts here, we conducted a landscape of instances of diagnostics that are approved and are in the pipeline, trying to address, to identify some gaps and priorities for R&D in the next 3 to 5 years. There are also the WHO diagnostic initiatives that is built around 3 pillars. One pillar is talking about R&D. Another pillar, for instance, is about capacity building. So there are several initiatives in the space to advance, hand in hand the development of therapeutics, diagnostics, vaccine, and try to contribute minimally to fight AMR with these tools.
Thanks again.
Thank you.
So now it's my pleasure to welcome Dr. Neha Prasad, again, an impressive bio that I need to read, and you're obviously a person in translation itself. I'm very impressed. You're a scientist turned strategist at Rubrum Advising, and that's telling all. You are looking and develop and execute go-to-market and market access strategies at Rubrum that align regulatory, clinical, commercial, and policy outcomes. And all that you will use to share with us hopefully a value proposition for NTAs. Very much looking forward. Thanks and welcome.
All right. Good afternoon, everyone. My name is Neha Prasad. I'm a scientist turned strategist. I started as a chemist at the bench in 2014. And as many of you have also realized, while science is definitely one of the barriers to getting new antibiotics to patients, another big barrier is, getting the approved drugs to the patients at the end after FDA approval and such. So this brought my career to turn in a very interesting direction. I started working with Lee Fleischer, who's the former Chief Medical Officer of CMS. CMS stands for Center for Medicare and Medicaid Services. So this is Medicare related. I had no idea what reimbursement was, how pricing worked, none of that. And they took a chance on me, and I started at the firm there to really learn about market access and apply all the insights I've learned there to the AMR field. So I've worked with almost 80 companies now across different modalities, therapeutic areas during my time at Rubrum. And this paper that I'm about to present to you on is really an outcome of that experience. I also want to call out Sumati Nambiar, who I collaborated with on this paper. She's the former director of the Division of Anti-Infectives at the FDA. She's been working on this for 14 plus years at the FDA itself, and she had really great insights that I wanted to share with you today. So right now, the way our situation is set up and why we're set up for failure right now is that we're really designing new antibiotics to fulfill this unmet need of treating drug-resistant infections. And rightfully so, right? That's the call we're hearing from ID docs, and that's the call scientists are responding to. But what this means is that it's directly at odds with antimicrobial stewardship, which says, hey, you should limit use of these agents. Don't use them up front because you're gonna perpetuate resistance against them too fast. And now that leads to low antibiotic utilization, which then historically has limited volume-based revenue. Now with revenue affected, you get this lack of return on investment, which limits future funding. And then you get no more antibacterial therapies, infections become untreatable, and then the whole thing, doom and gloom. This is the one slide that will describe the problem, and then now I want to move to potential solutions to escape this vicious cycle that we've seen play over and over again. So to start shifting the paradigm, the first step is, instead of this unmet need of treating drug-resistant infections, what if we thought about it in the framework of sparing antibiotic use? This is really similar to, for example, in the opioid use case, where in order to spare opioid use, new manufacturers are coming up with different pain therapies so that opioids don't have to get used. And you're probably asking yourself, why would a patient, why would a hospital, why does anyone care about sparing antibiotic use? Like, whose problem is this? And in the next few slides, I hope to convince you that Medicare does care. Wow, that should be a slogan. And then, yes, the rest of it will basically follow from there. Let me know if you have those questions at the end of this. Okay, so why does Medicare care? Well, because Medicare is the largest single payer in the US health system. For those of you who live in single payer health system, congratulations on not being one medical emergency away from bankruptcy. But for those of us who live in countries where there's many payers in the healthcare system, CMS is a large one that we think about. They set a lot of precedent that a lot of commercial insurers follow on, so on and so forth. So if you look at these two graphs, basically they show you that the biggest burden in AMR is felt by this geriatric population. Good, there's no pointers there. Up there on the left, on the right side one, and on the left, it's actually this bottom one. The graphs are inverted. Sorry about that, but. Yeah, basically, it's a problem in the geriatric population, and that's the population that CMS really insurers, right? And so now basically it's all aligned at this point, because if CMS is paying for the healthcare of this population, and this population is very sick, now CMS is going to be paying a lot more to treat that sicker population. But if CMS can get our population healthier, then their costs will also go down. So here the financial incentive lines up. And indeed, Seema Verma, who used to be the CMS administrator, already knew before those papers, really key papers were published, where she basically stated that, CMS is aware of this, and they actually did take action. And that action, one of them, is to promote antibiotic stewardship. So how did they do this? They basically used the CDC's core elements of antibiotic stewardship programs, which is this nice blueprint that different healthcare settings can use to facilitate antibiotic stewardship. And so CMS basically mandated that hospitals follow antibiotic stewardship programs. Now, it's not a legal mandate. It is a financially driven mandate because it's through this condition of participation, which means that in order for hospitals to receive payment from CMS, they need to show that they have an antibiotic stewardship program. And by the way, that condition of participation also includes an infection prevention and control aspect as well. And this had a lot of impact. You can see that there was a 50% increase in antibiotic stewardship programs between 2015 and 2020. So, hospitals obliged, this was in direct response to this new regulatory mechanism. So again, there wasn't any need for new legislation to be passed. This was a mechanism that already existed that CMS could employ to really make a difference in the space. All right, and then I won't talk about this too much, but my team had the pleasure of working with different hospitals and hospital associations on analyzing this condition of participation and finding room for improvement. And you can read more about this on our Rubrum Advising website. Okay, back to the plot line. The second step is, now that you have antibacterials that are fulfilling this unmet need of sparing antibiotic use, that means that antimicrobial stewardship practices will promote the use of these antibacterial therapies. So now you're no longer swimming against the current, you're swimming with the current. And like Valeria said, it's not a one-size-fits-all approach. It's really every different type of NTA will have a different value proposition, will have different regulatory framework and clinical trial designed to support all of that towards commercial success. So I have this chart here. You can also find it in this paper, and I didn't want to go into too much depth on it, but know that, if you're a NTA developer, find your road that fits yours and see if any of these options are ones you've thought of or consider implementing. All right, the third step is, now that you have antimicrobial stewardship practices promoting the use of these new antibiotics or NTAs, now you can get this high antibacterial utilization, which can drive volume-based revenue potential. And I know this is like taboo at this point because we've all moved towards Pasteur and delinking the revenue from the volume, but hear me out. In today's current existing framework, this is the way to go, right? Where you go for high utilization. And if you truly are aligned with antimicrobial stewardship practices, that will drive that utilization. Oh yeah, one shout out is, someone had flagged to me that hey, maybe you're still not going to get enough volume of prescription in order to make this work. And to that I say that, if we don't have these new stewardship guidelines yet that describe these new NTAs in this way. And once we do, I think we'll really see a difference in prescribing practices. And I also want to shout out this Vista contracting model, which we'll see very briefly at the end, which also tries to move away from this volume-linked model. But just for the sake of what's possible today, here's the volume-based model. So basically, now that those three top pieces we've discussed are done, the rest of the pieces really fall into place. And basically, this is kind of the way to turn things around. for us. And this all rests on the assumption that NTAs have a generally favorable safety profile as compared to standard antibiotics. And if that is true, then the rest of this logic does flow. And if it's not flowing, let me know afterwards. All right, so this is the VISTA model that I told you I'd give you a quick preview of. It's this new contracting model that I've been developing with my colleagues at Rubrum and others. But it's basically, to sum it up, a delinked pool incentive framework implementable through a scalable contracting model that does not require new legislation. So in simpler words, it's all the benefits of Pasteur without needing new legislation. It just it uses contracting as a mechanism to align everyone's financial incentives. Something like, in hospitals, the ED departments and others, radiology, all these follow this type of model. And so I do think that there's relevance here for the AMR space. So stay tuned for more on this. That's my next project. But yeah, I'll end here.
Here we go.
Hey, Dan.
Love your presentation. Love your positivity. I think it's great. The one piece, though, with the stewardship part that I'm not sure if it's going to totally align is the cost, right? Like, can we get away with charging $10,000 for a non-traditional, $20,000 for a non-traditional per patient? Will that model work? Right now, it looks like Solder, that is working for a small molecule, single agent, single bug, narrow spectrum. I'm hoping it'll work, but I don't know if it will work for non-traditional. I'm worried about the economics. The last part of your flow chart, I'm worried about that. When a hospital is telling their pharmacies, use vancomycin, it costs $20. That's the part I don't know.
Yeah, so it's interesting you bring that up because right now, yes, like the hospitals are incentivized. The way their reimbursement works is through the DRG, diagnosis related group, right? They get a specific payment based on whatever diagnosis that a patient comes in with. So you're given $20K to treat a patient for this other disease that has nothing to do with infection, but they contract an infection in the middle of it. And so now you get like a tiny little bump to account for that. But really it's all separate, right? And this is really the problem with the reimbursement framework right now is that all these budgets across the hospital, from the pharmacist budget to the physician budget to the actual hospital facility costs, all these are spread out. And that's not how it works in other therapeutic areas. The way it works is they've got service lines. I think there should be an ID service line, which is not an original idea. The IDSA has also put out a paper related to this, but I think that would solve a lot of the issues there, where you won't see that like transactional cost saving at such a, in a short-sighted way. And it's more, you're seeing the long-term impact. Because if you think about it, CMS is that payer who sees the entire costs of a patient population that has this, AMR issue from beginning to end. Whether you're talking about in like a clinic, in the hospital, in a doctor's office, at a dentist's office. And there's models that we're working on for that. So that's one of the integral parts of this model. Thanks for highlighting that.
Great, thanks. Looks like we're moving on. Thanks a lot.
I really thought more people would. Okay, thank you.
So we're starting having very interesting discussions here. It's not about the costs only, but also the development will be part of the costs. And so I'm very happy to have a regulatory view as a next speaker. So Dr. Giulia Genova is a medical doctor with clinical experience in cardiology. And she worked in research and teaching with the Swiss universities of Lausanne and Fribourg. So, Julie, you know all the stakeholders by experience, and so I'm very happy that you share your view on these questions. Very welcome.
Good afternoon, everyone. I hope to continue with the positive energy from NEHA, because as regulator, my task is a little bit difficult to have that energy. But I would like to start with the last slide from NEHA. She mentioned it's not necessary to change the legislation in order to promote access for that novel therapy to the patient. I agree, because to change the legislation is not easy. In Switzerland, to change the legislation takes between 10 and 15 years. And during that period, we have to adapt our procedure and to be able to promote the development of that product. We have not to wait to change the legislation because the process is very, very difficult. Who we are, Swissmedic is the regulatory agency for marketing authorization from all kinds of medicinal products. We are responsible for medical devices as well. And under our responsibility, we perform inspection in different establishments, and we are responsible for the market surveillances of the products. The pharmaceutical industry is very, very important for Switzerland because maybe you know, maybe we are not aware, but they represent more than 52% of the export of Switzerland. And we are responsible for that development. We have to promote that development and not to stop the development. And we are responsible to drafting the law, the legislation, and I mentioned we are not responsible for the prices. We are very, very happy not to be responsible for the prices because the reimbursement process is very, very complicated. That is the Federal Office of Public Health responsible for that. We have a strategy goal. Here I want just to mention that the most important strategy goal from Swissmedic is that one. I cannot show. That is the S3. Swissmedic support the development of novel therapeutic product and help ensure the Swiss access to innovative product. And what is the reason that I represent here that product? I'm responsible for advanced therapy medicinal product. There are gene therapy, cell therapy product, tissue engineering product, and the novel therapeutic, the NTA. They are not apart, not really apart for advanced therapy medicinal product. But we are only one division into Swissmedic to be all in one. Swissmedic is based on processes. That means we have department on quality assessment, we have department on clinical assessment, preclinical assessment, and the division of advanced therapy medicinal product is all in one. That means we have multitask experts. We have all together clinical assessor, preclinical assessor, quality assessor, and inspection. And that allow to have the possibility to have the contact very, very easy and very fast. And we can discuss in case of different product without, for example, enough clinical experience or clinical trial or preclinical data. We have the possibility to decide very, very rapidly if that will be possible to approve the product or authorize a clinical trial because of novelty of the product. And that is the reason to have such product as phages in our group under our responsibility, and microbiome as well, or CRISPR-Cas. CRISPR-Cas is another category that is clearly in the group of advanced therapy product, but we have the possibility to apply a very, very flexible procedure. That is the highlight of my message to you. We have the possibility, despite the law is clear. The law said we have the requirement to have the quality, the preclinical, clinical data in order to be able to approve the product, but we have the flexibility to decide risk-based, if possible, to approve different product. And here I can give some example. For example, last year we approved microbiome product, and that was based not of really randomized clinical trial. We approved that product on the basis of bibliography. We can accept that. And we have to adapt the requirement, the legislative requirement of the product in order to be able to give the rapid access to the patient. I mentioned assessment is risk-based, and that includes the inspection. We have the possibility, because our quality assessor, they can perform inspection as well, or together with our inspector. And they have the idea at the beginning and the start of the development of one product, what are the problem during the procedure of manufacturing process. And they can decide later with discussion between the preclinical assessor and clinic if possible to approve the product. Our position, for example, for phages, we can accept different design of the clinical trial. It's very important to be aware. We are on the moment that the phages, they are very, very important in the last time. There are a lot of discussion. Different countries, they are more developed in the application of that product. Another country like Switzerland, they start now. With the application of that product, but it's very important to mention here we are very, very open for discussion with the stakeholders. For example, if they are developer of that product, or if they are medical doctor, they want to apply that product, they have to come to us, and we can discuss with the different aspects, the necessity, the medical need, or the possibility to start a clinical trial. The clinical trial should not be randomized, and that is the gold standard for clinical trial. For such product like phages, that is not mandatory, because that is not possible to have a clinical trial, a randomized clinical trial with phages, because they are phages cocktails, they are personalized medicinal product. And to compare with a randomized, to compare as placebo or conventional therapy, that will be not possible in each cases. Just to summarize, we are open to discuss with the stakeholders different design, flexible design of clinical trial in order to move forwards with the application of that product. Here I want to mention we are not alone in Swissmedic, because we are working together with other authorities. We are very, very well connected internationally with in different platforms. For example, we are a part for Access Consortium, very, very important consortium between the countries outside the European Union, Singapore, Australia, UK, MHRA, in Canada. And that is a platform for us to discuss, to harmonize the criteria for approval of different products, or to discuss the criteria to be applied, but we have the possibility to assess different aspects. For example, MHRA from UK, they can assess the quality part of one submission, and we can assess the clinical part of one submission, and so we can harmonize our criteria and not to have the Swiss finish for into decision. We are part of different other products, for example, Open, or we participate in exchange of different information with guidelines with different countries. The international cooperation in the last time is very, very intensive, and that is very important for Switzerland, because Switzerland is a small country, and we have our legislation, but the product, they are not only in Switzerland submitted, they are submitted, for example, in Europe, EMEA, and after that they are coming to us, and our goal is not to stop that product, but to promote the development. We have innovation office. What is the reason to mention that here? That is a strategic goal for the Federal Council of Switzerland. And that allows to have different discussion with scientific advice, the stakeholder, the developer, the small startups. They have the possibility to come to us free of charges and to have a scientific advice meeting from the beginning, from the development of one project until to submission of for marketing authorization or for clinical trial. And we have already performed more than 130 such scientific advice meeting, and the feedback is very, very positive. And I mention here just in order to encourage the people in case of interest to come to us and to discuss the preclinical, clinical, GMP documentation, and so we can avoid problem early on, later stages of the development of one product. That is my last slide. Here are our coordinate. Please do not hesitate to contact us. I'm open for questions.
So being flexible is also a very, very optimistic way of looking at it. Thanks a lot. We are open for questions. As my fellows know, I'm always having one. Is it also, are we Swiss actually good in splitting the problem and still working with it under one hat? So you looked at all the NTAs we can imagine, but still under your roof. So is that the way to go?
It's a new strategy for the regulator. In the past, it was the regulator was very, very close, and we understood the thinking is different. We changed the culture on the thinking because it's clear for the product they are very, very innovative. They are very, very well advanced. It's not possible to apply the same criteria. We have to have a very, very intensive dialogue with the stakeholder and to exchange the experience, because a lot of that product, they are new for us as well, and we don't have the experience, and the developer, they have the experience. And we are very, very open to accept, if well argumented, new design, clinical trial design, new approaches, because our goal is to be a first wave for the submission for such product, and the non-traditional antimicrobial, they are a part for that novel product. That is the reason that we are responsible as Division Advanced Therapy Medicinal Product. Not our product, they are a part of the definition, but exactly that flexible way to approach the problems, to approach the procedure. It's new for Swiss Medic, and we are very proud with that.
Thanks a lot. And thanks again.
No question for the regulator. Thank you.
So, Eric, we go now to the proof of the pudding. My pleasure to welcome Dr. Eric van der Helm. He's a Vice President and Head of Business Development at Snipper Biome, where he connects scientific strategy with business development. That's a good way to go. He will tell us about his journey and is our example of today for fostering the discussions on NTA developments, and I'm sure you will show us all the pleasure. Thanks a lot. Welcome.
Thank you very much. I'll try to keep a positive vibe going. As Peter said, my name is Eric, representing here Snipper Biome. We're based in beautiful Copenhagen, Denmark. So what is Snipper doing? At Snipper, we're developing CRISPR therapies, and CRISPR therapies specifically targeting bacteria. So what you see here in this cartoon is, in essence, the core of our technology. We're trying to get rid of the rat pathogen, be it E. coli, be it Klebsiella, and we do that using CRISPR. So as you might know, I cannot point, but there's a CRISPR modality that can cleave the DNA specifically, sequence specifically. So that means that by programming the RNA that you see in red, you can very specifically determine where the CRISPR machinery makes a double-stranded break, which is lethal for bacteria, and which DNA it should leave intact. So all the commensal good bacteria, for example, in your skin or in your gut. We've worked on this for the last decade. This actually worked already quite early on in our labs. The key is actually how do you get these CRISPR systems into the bacteria? And that's actually a common theme when you also think about human gene therapy. How do you actually deliver the gene therapy to the right cells? And it's the same for bacterial gene therapy, as you could call this. We have two modalities. You can either deliver the CRISPR modality by living bacteria, as you see in the bottom. You can then deliver the CRISPR system by bacterial conjugation. Or, and that's the lead asset Sniper 01, you can deliver it using engineered phages. So in that case, you take wild-type phages and you use synthetic biology to, for example, change tail fibers and add CRISPR to the DNA. I'll show more in a minute. So this is our pipeline. I'll not go into great detail here, but I ask you to focus on the third column. So basically all priority pathogens, we're going after E. coli, Klebsiella, Pseudomonas. And we do like to, and that's shown in the last column, do things in partnership. So, for example, a big thanks to CarpX and Ed Burman, our alliance manager, for working with us for more than 5 years on moving ahead Sniper 01. We like to publish our findings. We think we're not operating in isolation. So we have published the blueprint of Sniper 01 in Nature Biotechnology a few years ago, and I'm quite proud to share that the phase 1 data that we ran 2 years ago is now also published as of this morning in the Lancet Microbe. So all data out there, open access for you to look at. And also talking about the points just mentioned about compassionate use, we're also using Sniper 01 IV in expanded access cases in the US. So zooming in on 001, Peter actually asked me to, because there are a lot of drug developers in the room, to share a few lessons. And I didn't know that this morning's session also started with a timeline 10 years ago, but it's, I guess good examples follow. So I'd like to take you quickly through the journey of Sniper and then highlight a few things that were quite transformative for a small biotech company. So first of all, we started out in 2015 based on patents. So we filed some quite early on some IP to show that you could use CRISPR to modulate bacteria while leaving good bacteria intact. That then allowed us very early on to raise a seed round. So this is important if you're, as an investor, you would like to invest in something tangible. So in this case, granted patents are quite a tangible asset. This seed funding then allowed us directly to go for the second catalyst, which is generating in vivo data. So with the seed round, we were then able to generate compelling mouse data that the technology that we written up in our patents and shown in the lab worked actually also translates to animals. And that again was a catalyst for us then to raise our Series A back in 2019 of $50 million from a consortium of VCs. Which then, and that's then the third catalyst, allowed us to work together with CarpX. And again, at that time when we applied to CarpX, it was very important to also show preclinical data, to also show that it actually works in an animal model. So again, this data is very relevant to push your pipeline forward. Received IND approval from the FDA, fast track designation, and then the last transformative moment was the first human dose trial that started the Sniper 01 in the US. Published our data, as I just shared, started working with the Gates Foundation, again working with CarpX on the 1B trial, started working together with the German federal government, and then also under, it was just mentioned, HERA, together with the EIB, provided us also with funding to continue the development. and very pleased to share that last summer we closed our Series B round led by the Cystic Fibrosis Foundation together with the German federal government. So this is just in a nutshell some highlights that allowed us to move forward with our development. Very briefly, what is Sniper 001? What is this non-traditional modality? It's shown here in this cartoon. And what we basically did is we started out with screening 162 wild-type phages that target E. coli, tried to find ones that were amendable for engineering, where we could swap tail virus that we could use to engineer a CRISPR payload into. And we ended up with 4 CRISPR-armed phages, as we call them, that we then also showed that in animal models worked. And then very briefly, the indication we're going after, it's a mouthful, but it's the title. It's the prophylaxis of bloodstream E. coli infections in patients with hematological malignancy at the risk of neutropenia. And then you might think, why? It sounds very specific. You could use an antibiotic in much more cases. That's true. But this represents a very straightforward, or not straightforward, but at least in our view, a clinical development pipeline that we think we can move forward in an efficient way. And the reason why we went for this indication is shown in this graph. This is data from Mike Setlin, collaborator at Cornell University. And what he found is that on the y-axis is the percentage of patients that receive a bloodstream infection. And he split up these patients before they received their transplant but shown in the gray bar, whether they were colonized with a fluoroquinolone-resistant Enterobacterales or if they were not colonized with a resistant bug. And that's shown in the white bar. And you can see that the difference is quite dramatic. If you're colonized, so you can just use a simple plate assay, and you're colonized with this fluoroquinolone-resistant version, the chances you get a bloodstream infection is 30%. So 1 in 3 of the patients that go into the hospital that you can actually screen for will get a bloodstream infection. And that's exactly the population we're going for with Sniper 001. Our aim is to push down that black bar down to the white bar, so you reduce the incidence of bloodstream infections. Very quickly, clinical development plan on the top, first human dose trial done, published in The Lancet this morning with beautiful dose response, as you can see in the middle, no safety events compared to placebo, and then we also saw some early signs of target engagement on the right-hand side. Phase 2, working on that, and then the phase 3. As you can see, the patient populations are not very big because you only need a safety database, at least at the FDA, for around 200-300 patients. So with that, our 1b/2a trial is currently going very well. 8 sub-sites in the US are recruiting since October 25, so we'll have a readout of this over the summer. Thank you very much.
Terrific, thanks a lot. We have questions, ready for questions, and I have, again, the privilege to... I would, of course, be very much interested. Do you remember some moments where you had some headwind or tailwind because you're developing an NTA?
Yeah, it's a good question. I don't think we and that also goes back to the previous points that were mentioned. We're not sitting in Copenhagen thinking, we're an NTA. Oh, in which bucket do we fall? I think, I think that distinction is not helpful. If you think about are there challenges that are specific for phage-derived products, such as the one we're developing, versus a small molecule? Yes, there are. I mean, for example, some CROs that we outsource our animal work to are reluctant to work with phages because it might contaminate the work they're doing for other customers. So I think there are challenges, but they're clearly solvable.
Okay. Again, another very optimistic last sentence. Thanks again. If there's no further questions, let's discuss in the next coffee break. And thanks again, Eric. Thanks to all the speakers and thanks to all the audience. And pleasure to continue to discuss on NTAs. Have a good afternoon.